Hello friends, so in today's lecture we are going to talk about a lytic cycle of a bacteriophage. So we all know that bacteriophage, it is a virus that infect bacteria. It is called a bacteriophage. It has two types of life cycles, a lytic and a lysozyme. So in lytic cycle, at the end of the infection, it lyses the bacterial cell and releases about 200 of phage cells. And in lysogenic cycle, it do not lyse the bacterium, but it is integrate its genetic material onto it, on bacterial genetic material. So if we saw the structure of bacteriophage, especially a polyphage, which infects the E. coli cell. So it tadpole-like structure. Here's the base plate, tail fibers, spike. Here's the sheath part. Here's the capsid. Inside the capsid, a complete packed DNA, double-stranded DNA material. This is the tail, and here's the core part. Here's the sheath. A sheath contains 24 annular rings, which works like a spring, ups and downs, pulls and pushes. This is the base plate, a hexagonal base plate is there also. Onto the base plate, there is 6 tail fibers, which is attached to the bacterial cell wall and penetrates the genetic material. This is spike. A sharp spike is located inside below the base plate, which is integrate into the bacterial cell to integrate the genetic material of phage into bacterial cell. So let's go on through the whole cycle. If these are the steps of cycle, there are mainly 6 or 7 rough steps, and clearly 5 steps is there. So the first step is attachment. The second step is penetration. Third step is early mRNA synthesis. The fourth step is late mRNA synthesis. The fifth step is phage component synthesis. The sixth step is phage assembly. The seventh step is bacterial cell lysis. So this rough 7 step which you can use to crack the whole cycle. So let's go on through the whole cycle, what actually happens. So this is the bacterial cell, E. coli. It is a bacterial genetic material. So what happened? The bacteriophage, it attached on the surface of bacterial cell with the use of tail fibers. Now the attachment is very specific because in the tail fibers there are specific ligands out there, and these ligands bind to the specific receptor present on the cell wall of the bacterium. The receptor may be onto the LPS, maybe onto the fimbriae, or maybe onto the tail. So after the clear attachment, the bacteriophage sheath, the middle sheath is pushed and it is bent. So the sheath is contract and the bacteriophage is pushing down, and it pushes a spike penetrate onto the bacterial material. The bacteriophage is now pushes himself down, so the spikes gone to the inside the bacterial cell. The sheath is contracted and the phage genetic material gone into the bacterial cell. So now what happened? The remain phage after the integration, this is phage genetic material, sorry, bacterial genetic material, this is phage genetic material, and here the phage. A genetic material less phage because only a phage genetic material is penetrated or comes into the bacterial cell, not a whole phage. So the main phage is called G host, or it's also called dominant phage. Now we do not have any need. So now after the integration or penetration of this genetic material, what it do? There are two types of genetic material, right? A host genetic material and a phage genetic material. So what commanding the cell, the right, the E. coli genetic material. So what phage actually have to do? It's command the cell. So how it command the cell? How it control the cell to degrading the opposite genetic material. So what it actually do? It synthesize the early mRNA. In this early mRNA, it transcribe or translate the specific types of nuclease. So what nuclease do? Nuclease do is break down the host genetic material. Now you have a question that why it only break down the genetic material of E. coli, not there, because it is a nuclease. It do not have specification. So into the bacterial fast DNA, there is a cytosine, but this is not a simple cytosine. It is bound with the hydroxymethyl. So due to this, the nucleus is clearly identify which material is on and which material is opposite. So it breaks down the E. coli genetic material. The E. coli genetic material is now fragmented, and the whole control of the cell is now under bacteriophage DNA. It control the cell. Now what happened after the control? What it do? It just increase their population. So what actually they do to increase? It simply synthesize its own phage component and also replicate their material. So next is synthesis of late proteins. Now what late proteins do? Late proteins do the simply the synthesis of phage component. Ultimately phage component synthesis. Now we also don't do the fragmented DNA. Now this is the phage DNA. It replicates. And the phage component is synthesized. This is the head, a base plate and 10 fibers. So after the synthesis of phage component, what it do? It assembles all together. Now, assembly of phage. Approximately 200 phage is assembled, or 200 phage is synthesized per bacterial cell to lyse the bacterial cell. After the 200 phage is synthesized, is generally what do it do the lysis of bacterium by pouring. Now it's possess the pouring protein. What we do in lytic protein, we generally pour or the cut the cell walls a simple creating a simple pore. So what did it lyse or it bombs the cell and release the approximately 200 phages. So release of 200 approximately 200 phages. So this is the lysis and release of approximately 200 phages. So this is all of the lytic cycle. What we actually saw that attachment of bacteriophage, then penetration of genetic material, then taking the control of genetic by breaking the opposite genetic material, it takes the control of the whole cell, then it synthesizes all component, then what it do, it assemble the whole this bacteriophage, then it lyses the bacteriophage. I hope you understand. I hope you like the video. If you want to learn anything from me, comment in the below the comment section. Stay connected with Ravi's Microbiology. Thank you.